well-known evangelical preacher, and he left not only Christianity, but he became an atheist and opposed to God. And in this book, he talks about why. And there are a number of reasons he gave as to why. One is evolution, which I won't cover, but the other one is basically, you can see his audience there is huge. So he has quite a following. And basically, how could a loving God permit, much less create, pathogens? And that was a major concern that he talked about in his book. He just could not understand how a loving God could create all this stuff that's bad. And most people, when you talk about bacteria or viruses, of course, they are bad. When I say you got to wash your hands to kill the bacteria, you wash your hands because you know you have to kill the bacteria. Soaps advertise they kill 99.99% of the bacteria on your hands, and of course that is a selling point for your hands. And Templeton said, I'll just read word for word so you understand where he's coming from, the grand design also includes the creation of parasites, worms, fleas, ticks, lice, bacteria, viruses, and the aberrant cells that attack the body, the organs, or brain, and the malignant cells that cause death and suffering. The latter part, of course, is cancer, and I won't cover that now, but I think I will in the future because a lot of people misunderstand cancer and what causes it and why it's a problem. Charles Darwin himself asked, why did God allow evil to exist? If there's a loving God, why did God allow evil to exist? You read books about philosophy, one of the major areas they cover is, why is there so much evil? And they go through this. We have a God that allows evil, or causes evil, or God's not all powerful. Lots of reasons are given, which is a area I won't get into now. Genesis, God saw everything was very good. And the important thing we have to keep in mind is Genesis' fall caused corruption that affected the entire creation. Thus, we expect an imperfect world, and I'll mention in a couple of cases where that is true. Where are bacteria located? It may surprise you that you have billions of bacteria on your body, namely in the gastrointestinal tract and the surfaces that have contact with the outside environment, skin, upper respiratory tract, and the genital tract. The rest of the Bible is sterile. Should not be bacteria. Okay, 99.99% are good, which I'll detail in a minute. Harmful bacteria are really rare. Really rare. And we now know, and I just got a case history from somebody a couple of days ago, to be healthy, you need to be exposed to bacteria. And the best example is young children. There's a puddle here, it's dry here, it's dry there. They will go out of their way to walk to the puddle. They need to get dirty, it seems. And the reason they need to get dirty is that they need to be able to develop the immune system, and that means early exposure to bacteria. In fact, too clean is a problem, and the best example is we had a nurse who autoclaved the sheets for her son. I don't know, people ask her where she got the autoclave, I don't know, but she autoclaved. She wanted to raise her son in a close to as possible sterile environment. That child, when that child was about 5, died because he never developed the proper immune response. So you need to be exposed to bacteria to develop that response. Now, babies, when they are born, they are sterile. So how do they get bacteria in their system? One way, a major way they do, is God created us to have a perception that that little guy is cute. And if you disagree, there's something wrong with you, right? I mean, babies are cute. Even politicians will kiss babies. And so therefore, they are regarded as cute. So what happens? You kiss the baby on the cheek, on the forehead, on the hand, whatever, and those bacteria then take a vacation and they travel from the cheek into the digestive tract, and within 2 or 3 days they populate the whole digestive tract. So that's where it comes from. So I think, and I see this when I'm doing research, over and over you can see it's God's design. This is how the bacteria get in the system of the baby, face, the attractiveness is we are designed to respond to that. So it's all design system. And what about kissing or families? Well, when you are dating somebody, your bacteria becomes hers and vice versa. So after a while, there's no problem. Promiscuity now, you end up with so much foreign bacteria, your body can't often handle it, and therefore you get sick or could get sick.
Dr.
Family, same.
Could you move just so, yeah, right about there.
Family, the same thing. The family, of course, the bacteria is shared among members, and therefore kissing's okay. If my baby eats out of a spoon, I can eat out of the spoon. We trade bacteria, and everything works out really, really well. So why do bacteria become pathogenic? We know this. I taught microbiology. There's no question about what I'm covering. And number one reason is mutations. You have a mutation, a mutant form. The E. coli is the best example. You have a mutant form, and that is a problem because it has a mistake, a change that is problematic. E. coli are not normally problematic, but certain strains are. Another is plasmid transfer. Bacteria has plasmids, so small round DNA sections, and this can be transferred from one bacteria to another. They do this quite often. It's a way that bacteria can get more genetic variety, and in doing that, sometimes you get a mutation that is transferred to another bacterium, and that causes a problem. Another problem is a weak immune system, and that means if your immune system is strong, you typically don't have a problem. If your immune system is weak, as with COVID, a good example is they stress they should be put masks on 5-year-olds and go through all these fancy protocols. And I have a master's in public health, and one thing they stress is you need to be exposed to these bacteria when you're young, so you develop resistance to it, and that stops or slows the spread of the bacteria. And the best example is during COVID, I had spent a week in Indianapolis at Christian schools teaching, giving presentations. And curious, because nobody wore a mask, no one seemed to worry about that. Many of the other schools were closed at this time. And so I was curious, and I talked to teachers, parents, and so on. What about COVID? We haven't had any problems at all. No masks, no concern. Yeah, your kids, they concerns, but it's not a problem. One parent said, I got COVID, and I kept my child home from school for a week. I got over it. Child never got it. Back to normal. It was the only case that I was confronted with of someone who got COVID. And now we know pretty well that the problem is not young people. To try to get young people to wear masks is irresponsible. And it's primarily people who are affected by COVID are those with a weak immune system. Most all had a comorbidity, and that's why they ended up with a problem, and many people died. And some I thought were healthy died of COVID. I asked, but I thought so-and-so was healthy. What was the problem? Well, but he had a lot of problems. He had this and he had something else. It didn't surprise us when he succumbed to COVID, and therefore we recognize that weak immune system, in this case, was part of it. Part of that is age. The immune system at age 50 is half as strong as it was when you were at age 10. So the immune system deteriorates with each age that you get. Each year you age, it becomes slightly weaker. Now, it doesn't have to be half the strength, but on the other hand, on the average, it tends to be. And so therefore, older people need to be protected more so. Compromised immune system is another problem, like AIDS. The virus attacks the T cells, which is part of the immune system, and therefore it is compromised, which is a problem. Number 4, a major problem is you get an organism in the host, and all organisms have a host. That means no problem. They live in the monkey, for example, like HIV. They're happy, no problem. Monkey's happy, no problem. The problem is when that, in this case AIDS, is transferred from the monkey to a human or from a bat to a human, then we have a problem because they are not in the host that they're compatible with. But if they are, they actually can be beneficial for these others. Okay, good example of Staphylococcus aureus, which is skin bacteria. On the skin, it's not only not harmful, but it's helpful, because it's like, best example is you have apartments, and you've got a lot of raunchy people in the neighborhood. How do you keep them out of your apartments? You rent all the apartments to good people. They're all paying the bills, and when the bad people come in and say, I want an apartment, you say, I'm sorry, they're all filled. No problem. Bacteria is the same thing. When they fill up the area in the skin, when pathogenic bacteria get on the skin, there's no room. They can't survive, because the good bacteria, like Staphylococcus aureus, has filled up the apartment spaces, so there is no problem. But when you get a cut, then the Staphylococcus aureus ends up in the body, then you have a problem. You can end up with a pretty bad infection. Okay, the mutant form, Escherichia coli, 015787. That's probably one of the most infamous strains of mutant bacteria, and that can be lethal in a week or 2 or 3. And if they find out your person has that, often from eating uncooked hamburger or other foods, if you have it, you got to treat the person right away. If you wait too long, you lose the patient. So that's very, very damaging, but we know that's a mutant form. Normally, E. coli is not a problem. And so that is an example where it's not, but becomes pathogenic. Also, they can acquire virulence factors from plasmids, transposons, or bacteriophages. So again, they can be transferred from the bad bacteria to the good, causing the good to become bad. And that, in case you haven't seen it before, that's Staphylococcus aureus. Very well-known bacteria. When I talk microbiology, you get a petri dish out, you put a medium on there, you put your hand on the medium, you take your hand off, you put it in the incubator, and within 4 or 5 days it's covered with these guys. Now these are colorized because most bacteria don't have color. You can't see them. So you stain them to help you see them, and that's what's done here. You use a number of stains so you can better visualize them. Okay, also, another example would be bacteria in the appendix. Should be there, no problem. But if it's ruptured, it can get into the peritoneum in parts of the body it shouldn't be, and therefore cause a problem. And I mentioned the skin bacteria, Staphylococcus aureus is a problem. Now, when I first took microbiology many years ago, the professor said, bacteria are your friends. And I thought, is this guy okay? My friends, come on, they're bacteria, they kill people. And he spent much of the course showing, indeed, that bacteria as a whole are our friends. They're important for our good health. And these are a couple of examples. Probiotics for health, the probiotic here. Many other examples, because now we realize the world has caught up to my professor's ideas. And now we realize that, indeed, you need bacteria. And I just had some yogurt today. One reason I had the yogurt was the yogurt has lots of bacteria in it. And therefore, I'm eating bacteria. In fact, you can buy called root beer belly, and root beer belly is just little kernels of bacteria. You can just swallow the bacteria, and the bacteria tastes like root beer, which is why they call it root beer belly. But it's a good way of getting bacteria in your body to help replenish the amount there. And areas where you need to be concerned about this is when you take an antibiotic. The doctor should always tell you when you have an antibiotic, it kills not just the good bacteria, it kills not just the bad bacteria, but the good bacteria as well. So when you have an antibiotic, penicillin, then you need to eat yogurt or some way of getting the good bacteria in your body. Okay, some sources. Yogurt, excellent source. Sauerkraut, apple cider vinegar, apples, olives, pickles, garlic, and there are many, many others as well. And this right here you can see bacteria again, and this has got the blue stain, and that stain is the same stain they use to stain blue jeans. And that's why they got the stain, because it really is a good stain. It stains blue jeans very effectively, but it also stains the bacteria. And so that way we can primarily see, you can see it. They're hard to see. And you have typically dirt and other stuff in there, and so this helps you determine what bacteria are there. And also they stain differently. So when you stain it, some bacteria accept certain stains, others don't. so that way you can determine whether or not you have the bacteria you're looking for. But when you teach microbiology, you see this sample here just about every day, so you get used to it. Okay, probiotic functions. What do they do? Well, one is candida, yeast infections they can help control. If the bacteria are in certain areas, the yeast is not able to get a hold of that area and reproduce. Manages digestive health, of course, clear skin. One problem with skin is we wash it too much, we kill too much of the bacteria, and as a result the skin is not healthy. So we need to use some lotions that actually replace the bacteria that we remove from washing. Reduces food poisoning because it breaks down certain foods so that poisoning is less of a problem. Immune system health, which I'll mention in a minute, controls nail fungus, again because of the yeast infections, and kills other pathogens. Why bacteria are important? Degrading food, of course, neutralizing toxins, especially vitamin K, produces vitamin K, thiamine, folate, biotin, riboflavin, cobalamin, niacin, protophenic acid, pyridoxine, and protophenic acid, synthesized, I think I messed up on one of those, by the gut bacteria. And so those are some of the things that they do. Without that bacteria, the only way you get a lot of these are through vitamin supplements or other means. And generally, bacteria is the best source for these compounds like thiamine, folate, for example, and therefore is the best place to get it. Also, they help with bacterial metabolism. They literally help break down protein and lipids, so they actually digest it in order to facilitate our digestion. And they make enzymes needed for vitamin B12 synthesis. And so without this bacteria, the only source of vitamin B12 would be supplements or a lot of milk products, and sometimes meats and fish carries the B12. Also helps maintain reproductive health, and unfortunately, we tend to focus on destroying bad microbes, and more health-involved people are trying to focus on not destroying bad, but encouraging the good to develop, because that tends to reduce the problem with the bad. And that is a nice three-dimensional picture. I think that's done with scanning electron microscopy, which is one of my favorite tools when I was at the medical school. Well, I tell you, I love to go out and catch spiders, and insects and bugs, and put them under the SEM, and just you see things that you would never see before. I understand ICRs can have an SEM soon. Oh. Okay, well, encourage it. It's really a fun thing to work with. How it works is incredible. Okay, they serve humanity, bacteria does, by making ethanol, enzymes. A lot of drugs that we buy from the drugstore are made by bacteria. So the factory involves bacteria to make the drugs. Best example, which I'll mention, is making beer and wine. When I was doing doctoral work at Miami University, they stressed the practical approach to chemistry. And so among the places we visited was a company that made beer. And they spent a good deal of time talking about the microbes. And you've got to make sure you have the right microbes. You grow them, mutations, big problem, and therefore if you end up with the wrong microbes, you end up dumping thousands of gallons of beer in some farmer's yard. You have no choice. So, and they have tasters, which I was able to try out for, and I failed. Tasters will taste the bacteria actually, but they'll taste the wine or beer and the bacteria. The bacteria produce certain flavors, and then they will determine whether or not it meets the standards. So after they produce 10,000 gallons of beer, the last test is the taster. It has to pass before they can put the beer on the market. And the reason is we are very used to a certain taste. If you're a beer drinker, I'm not, but you're used to a certain taste. And then if you buy a case of beer and it doesn't have that taste, that can be a problem in selling the beer. Cleans up oil spills, toxic waste, kills plant pests. One of the main areas which I was working in was gene therapy. Gene therapy is where viruses, especially, were the mechanism to transfer genes from the lab into a cell. And so we did a lot of work with gene therapy, especially lambda virus. Lambda virus was one of the major means, because it's a large virus, you can pull out a lot of genes and it doesn't kill the virus. Actually, you can't kill viruses because they're not alive. They're gene machines. But on the other hand, what do you call them? They're active, so I guess you can call them alive, but technically, that's not true. Now, sewers is important. John Snow, in London, they had an epidemic of cholera, and they could not understand what was causing the epidemic. And so he basically got a map, charted all the cases, and noticed all the cases, except one or two, were around the Broad Street pump. And so his suggestion was, to stop the epidemic, was put a lock on the Broad Street pump. And they thought, nuts. Why? Didn't understand. So, I'm not sure, but try it. Had nothing else. No one suggested anything else. So they locked the pump, they put a lock on it so it couldn't be used. And then the cholera epidemic disappeared. Within a week or two, cases were down to 0. Before that, there were quite a few deaths from cholera, and that's an awful disease to have. I would not recommend it. Given a choice, I'd pick something else. A choice. But now we know what happened was there was a sewer line that was cracked. The sewer was leaking into the water supply, so when they pumped water, they ended up with lots of bacteria in the water. And the taste evidently didn't change that much, but the cough up here, talking all week, so the taste didn't change that much, but it was there. And that's how they end up with cholera. Okay, moving on. Deuteronomy. There are many scriptures. In fact, there are a number of scriptures that talk about this, and I'll just read my translation of the King James. Carry a shovel with the rest of your gear when you have relieved yourself. Use it to dig a hole, then fill it, covering your excrement. And the Josephus works say, make the hole a foot deep with a spade. And to everyone a pickaxe is given for that purpose. So now we know, of course, why this is true, because a major spread of bacteria is a result of fecal matter. And it may surprise you that fecal matter dry weight is about 50% bacteria. And so in fecal matter, you're talking about trillions of bacteria. Most harmless, but a few are problematic. And therefore, if this rule was followed, there would be a lot fewer epidemics. Well, you wouldn't have had the Broad Street pump issue either if this rule was followed. And so what happened was the Jews, hopefully, most followed this. Therefore, when epidemics came along, they were less susceptible, and as a result, the non-Jews were claiming the epidemic was caused by the Jews. So they ended up being blamed because they followed this rule. And there are other examples as well. Quarantine. There's 16 verses in the Bible which talk about quarantine. Quarantine is one of the most effective means of reducing disease spread. If I have some disease, chances are pretty good people close to me will be exposed. And therefore, quarantine was used way back in Bible times. 16 verses is a lot of recommendations relative to quarantine. And again, the Jews tended to follow this. The Gentiles didn't. And as a result, Jews were less likely to get sick with these diseases, and therefore non-Jews were apt to blame them. Well, this is your fault. You're not getting sick. We are. What's going on? Now, what about toxins? Well, the best example is dose is the poison. Nothing is toxic in small enough amounts. Everything is toxic in high amounts. A good example is without water, you die. You have to have water. So much water, 8 glasses a day, I think they recommend. So much water you have to have. But too much water, and you end up dying. It can be lethal because it changes the pH of the blood, the electrolytes is messed up, and as a result, you can die. And The best example of that was a contest. People who could drink so much water and not pee. So the goal was who can drink the most amount of water and not pee within, I don't know, an hour or two or whatever. And one woman, she drank lots of water, she controlled excretion, and she died. And nobody, radio station, could understand why she died. Well, now we know, I mean, study biology, you know very simply the electrolytes were messed up. And your electrolytes are critical to keep those in proper balance, and that doesn't. So even though water is necessary for life, in high amounts, it is toxic. What about arsenic? Well, arsenic is a good example, and I'll give you one that was given to me at medical school in my toxicology course. There was a case of a princess who married a prince, and rumors were the prince was fooling around. These were confirmed, and so it wasn't long after this, and the prince died. And they did an autopsy, and they found high amounts of arsenic in his body. So she was tried, convicted for murder, because they concluded, with good reason, that she poisoned her husband, and that's why he died. Well, on her deathbed, she said, I have a confession to make. I did kill my husband, but I didn't use arsenic, I used strychnine. And that caused the scientific community to say, wait a minute, what? Why? He had arsenic in his body. That's logical, that's what killed him. Well, we know that all of you have arsenic in your body. That's not the issue. The issue is how much arsenic you have. And in this case, probably his arsenic level was a little bit higher than average, but we don't know now, but it was high enough that the autopsy on the prince indicated there was a level too high. So now we realize that the dose is the poison. And one thing I got from toxicology, our textbook was this thick, and I was in Sweden at that time doing lecturing, and I took the book along and tried to read it in Sweden, but any rate, learned a lot about toxicology. And that's one thing I'll never forget. I guess if you remember one strong point in a course, I guess that's well worth it. Okay, leeches. Leeches, well, that view is gross.
Yeah.
That's not a very photogenic picture of the leech. Profile's a little bit better, but not much. But not much better. But I've given this presentation several times, and so I had a number of testimonies from nurses and doctors. And I can add what they said when I presented this material. Leeches attached to the body, they live off blood. Well, so what? When you have a body part that's lost because of a graft or a finger that was lost and you sew it back on, it improves blood circulation after grafting or tissue reattachment surgery. And it does that for a number of reasons. Besides improving circulation, it releases a powerful blood thinner which reduces the blood clotting problem. It also releases compounds which reduce the pain. And so in many ways, it facilitates the attachment by better circulation, reduces the pain, produces antibiotics, and also reduces the problem of blood clotting. So it's perfect. That's all you have to do. And my nurses that were in the audience mentioned that, yeah, we have a jar of leeches, and when we have a patient that has reattachment surgery, we put the leech on, and the leeches are sterile, she said. We put the leeches on, the leeches do their job, and we pull them off every other day and put on a new set of leeches. And after a week or two or three, the attachment's successful, and the patient can go on with his life. Otherwise, most of these reattachments would not work. And so it's not that, you don't have to start giving them drugs and shots and so on. They do everything. So my guess is that God created leeches just in case we did something stupid and lost part of a finger or other body part. He's got the remedy right there. It's all set. All we have to do is put the leech on, nothing else. And we don't need to start giving them shots and antibiotics and all the other stuff. Just the leeches, they do everything. We're all set. So why did God make leeches? Well, my conclusion. Oh, Perudin is the anticoagulant that they use, and also, of course, analgesics. And can also treat body ailments like inflammation, certain skin diseases, rheumatic pain, etc. So there are a number of issues. How about maggots? Maggots are flies, worms, fly eggs they lay on decaying meat. And ironically, this is also another example of a really great surgical remedy for problems. Once surgery is taken care of, or in the case of diabetics, when you have tissue that's lost, the leeches, maggots, in this case, consume only necrotic tissue. That's all. Healthy tissue, they ignore. So you put the maggots on the area, on the foot in the case of diabetic injury, and then they remove all of the necrotic tissue, all the good tissue is left, and they also, again, add anticoagulants and antibacterial compounds and so on. So the maggots, all by themselves, take care of the problem. You don't need to do anything else, except change the maggots, I guess, once every other day. And when you present this option to patients, okay, we may have to cut the foot off or more, but you want to try maggot therapy? And most patients, when it's explained, say that's gross. And when they do it, a slight tickle, it's nothing. And they walk out of the hospital, both feet, and are happy that they utilized this option. And, that's actually known this for quite a few years. Surgeons noticed that soldiers that had maggot-infested wounds, their wound healed faster than infestation-free soldiers. Medical conditions they're used to treat, diabetic complications, I mentioned that, and primarily there are other diseases which cause damage to the skin and the tissue. So again, I think that God, when he knew we would probably have certain health problems, so he created maggots so that when we have these problems, we're all set. Okay, I mentioned this. Burn and accident victims, that's important as well for maggot therapy. Even osteomyelitis. Osteomyelitis is where the inflammation is inside the bone. I guess they get inside there and they take care of the inflammation as well. Mosquitoes, they certainly can't be beneficial because obviously they spread malaria and cause other problems. But actually, it's only the females that are the problem. Many insects, it's only the females that cause the problems because females lay eggs and therefore they have to have the nutrients, etc., to produce the eggs. Male mosquitoes, they suck nectar off flowers just like bees. So there's no difference between mosquitoes, male mosquitoes, and bees. Well, the flowers they attack, there's a difference. And actually, most insects are helpful to humans, including mosquitoes, at least not harmful. So what do insects do? Well, the biggie is pollination. Pollination is critically important. Without insects, you have to pollinate by hand. Good luck. That's our dubious job to start lifting the pollen from one flower, put it into another flower. Dispersing seeds is a function. They recycle dead plants, animals, and dung, and they control plant and animal populations, and they provide sustenance to other animals. Many animals are small, and they can only consume other animals that are very small, like mosquitoes. And so it's kind of a way between the really small creatures and larger creatures. So it's an important source for many larger animals. And then the larger animals, of course, are important source of food for other large animals. The difference, in case you want to look at the mosquito and see if you should swap, is right here. You see the arrow there. The male has these hairs coming off the twinges here, and you can see the female doesn't have that. And I'm not sure what that is for, but that's the difference.
Mustache.
Males are smaller than females, and as I mentioned, male mosquitoes feed only on plant juices. They get the sugar. Nails don't bite. Mosquitoes are an important means of controlling rodents, rats, mice, and rabbits. So they're really, for many farm areas, they're an important means of dealing with the rat population. And also, as I mentioned, they bridge this gap between fish and the near microscopic organism. So basically, you start out with small microscopic organism, and then you work your way up to whales. So each level has a preference for food, which kind of fits the fish or animal lifestyle.
How do they control rats and mice and?
Well, other ways of controlling rats, of course, like cats.
No, I mean, how do mosquitoes do that?
Oh, mosquitoes do that by affecting the females bite the rats, and then they affect the rat's ability to reproduce. So that's probably not a prominent source of rat control, but most of us just have a cat. That's the easiest. Spiders. There are very few spiders that cause a problem. Most spiders are benign. Some, like a black widow, it can bite, but deaths from a black widow bite are very, very rare. So they do bite, and the way you can avoid a bite is when you start to move rocks or stuff on the ground, watch yourself. Wear a pair of gloves. If you're moving something around on the ground, and that's where they live, that's where they subsist themselves on the ground. And they're, of course, pretty prominent because they have that hourglass, the red hourglass. And with black widows, again, only the female bites. And the primary diet for spiders is insects. In fact, most farmers, when they have a spider web, just ignore it. Because the spiders are an incredibly effective way of reducing problematic insects. They're not selective. Small insect gets trapped in the web, it's gone. No problem. And moving on. All creation is very good. When I read this, some people say, wait a minute, what about viruses? Viruses do cause a lot of diseases. No question. Viruses certainly could not be good. Well, guess what? National Geographic says they kill us by the millions, but without them, life is impossible. And if the National Geographic said it, it's got to be true. So it must be true. And it is. The reason that it causes a problem, again, is it gets in the wrong organism or organ. HIV from monkeys, no problem. To humans, problem. Flu from swine or ducks, no problem. To humans, a problem. Ebola, bats, no problem. Humans, a problem. And so the major problem, or a major one, is it ends up in the wrong host. And virus cause diseases, polio, hemorrhagic fever, diarrheal disease, smallpox, flu, HIV, Ebola, hepatitis A and E, and many others. There's a lot of diseases caused by viruses. Smallpox is probably one of the best known examples, and now we've pretty well, I think, totally eliminated smallpox because of vaccination. Campaign which ended in 1980, so that was the end. So the first disease ever eradicated, and it's gone forever. Okay, when you look at, in this case, AIDS, viruses, you can see that they're designed specifically to do some tasks. And inside of the virus, you have a coat on the outside. Then you have viral DNA on the inside, the capsid for this particular virus, covering the DNA. And then you have these membrane things that extend from the cover. In this case, you can see the yellow would be HIV, and the blue would be the other cells. And what do viruses do? When I present this, a number of people say this was the best part of the presentation. So what do they do? One of the main things they do is keep bacteria in check. Bacteria would, if they had the resources, the food and the room, they would cover the earth in a couple of days. And the best example we use in microbiology, if you take micro, you'll hear this over and over. But the best example is there was a servant who pleased the king so much the king said, "What can I do to pay you for what you've done?" And he said, "Well, bring a plate with 1 grain on day 1. Bring another plate with 2 grains on day 2. Bring another plate with 4 grains, day 3, 8, 16, 32, 64, etc. for a year. That's all I ask." And the king said, "That is nothing." Well, in fact, what happened was the man, who was pretty smart, and that's why the king was rewarding him, but basically, after a year, he had every piece of grain in the whole area. So he had monopoly on the grain. Because if you start doubling after 360 times, well, 365 factorial, you end up with a huge number. So that's what happened. And I'm not sure what the king did, but he probably realized that this guy was really smart, and maybe I should have suspected something, but he didn't. Okay, what are they? Viruses are called gene machines. They have protein shells, or they capsid, they call it. They're covered with markers. They're called antigens. And these markers basically are like a license or a ID tag. It tells the body what the virus is doing, what type of virus it is. And viruses are designed to utilize the basic cellular machinery. That means that when viruses get in your body, they use your ribosomes, your tRNA, all the translation factors, etc., to produce new viruses. And so that's what AIDS does. It gets in your cells, T cells, and it uses your equipment to make millions of new viruses. So they reproduce very, very fast. And the intriguing thing is they can't get into any cell. In this case, the HIV, it has to have 2 keys, CD4, which fits the CD4 receptor, and a CCR5, I think it's over here somewhere, receptor. So I have to have 2 receptors. That means that if you want me to go to your house and pick up something you forgot to bring to the studio, you have to give me 2 keys. So to get into your house, I have to have both keys. And with the virus, the HIV, it has to have both keys before it can get into your house. Okay, here we go. It's the CD4 and the CCR5 co-receptor. So it has to have both of those. Only then can it get inside of a cell. And that leads one to believe that viruses are designed to enter only certain cells and not other cells, because it has to have that fit. If it doesn't have that fit, it can't get into the cell. And one of the most interesting ones is the bacterial phase, T4, and there is a picture of the actual virus. You can see there's a drawing of the virus. And that is of special note to me because there was a man about 30 years ago who was doing research in this area, and he saw bacterial phage, and he said, there must be a designer. His name was Michael Behe. And that started the whole intelligent design movement looking at that virus. And if you think about it, you look at this thing, it cannot survive unless it has every single part. It cannot spread unless it has the neck, the DNA, the head, tail sheath, tail fibers, pins, end plate, etc. And there's an actual photograph of a bacterial phage. And so he realized that it's not going to exist until it has all the parts. And he couldn't imagine getting all the parts by any process such as mutation, because it doesn't have DNA except in the head, and that DNA is designed for a specific purpose. And there you can see a major function of bacterial phage is to destroy bacteria. And you can see there's a bacterial phages, and it's getting inside of the cell. And so cells don't have a chance when they decide to attack. They are finished. And that is one major way that your body can reduce the problem of bacteria infection. Okay, moving on. So we use bacteriophages to treat bacterial diseases. In fact, in many countries, the treatment for bacteria infections was a bacteriophage. We don't do this treatment anymore because now we have antibiotics. But if a person is resistant to every, or the bacteria is resistant to every antibiotic, the natural response is use bacteriophage. And so this, for some reason, hasn't been thought of. So you end up with people who are resistant to every strain. Just use a bacteriophage. Because I've never heard of resistance to a bacteriophage. I don't think it's possible. So therefore, just use a bacteriophage and you are all set. In fact, it is used in many countries still today. Russia, Georgia, the country, and Poland still use it. Some doctors feel that the problem of resistant strains of bacteria could be ameliorated by using bacteriophages. And I don't see why it couldn't be. I think it could be. But viruses are everywhere. 1 times 10 to the 31 power are estimated to be on the earth. Hot environments, cold environments, salty environment, inside earth, outside of the earth, hot, dry, wet, dry, doesn't matter. Even Egyptian tombs, and they found the viruses in the Egyptian tombs when they were released and got on people, even though they were sitting there dormant for 3,000 years, they caused viral infections in people. And so, as far as we know, besides from radiation, they can probably last for 10,000 years and still be functional. They have to be physically damaged not to be functional. And in your body are trillions of viruses, and they're all doing their work with nary a complaint. Why do we know much about this? Because in doing research, you have to have some reason. And when I did cancer research, that's easy. You're raising money. Why? We're researching cancer. What causes cancer? How it can be treated. People are willing to donate lots of money to cancer research. And often they said, yeah, my mother died or my aunt died of cancer, so yeah, I'll be willing to donate the money. Granting agencies are willing to give you the money to do research. What about the viruses in your eye? You have 6 different viruses. What if I knocked on your door and said, I'm collecting money to do research on the viruses that are in the eye? And you'd say, well, what disease do they cause? Well, we don't know of any disease they cause. We just know they have to be there. But what do they do? I don't know. Look, I'll give my money to someone doing cancer research. I really don't want to give money to satisfy your curiosity. I mean, I'm glad you're curious, but I'm not interested in you trying to find out what those viruses do. What do they probably do? Probably what they do is kill bacteria, but we don't know. We just know if you don't have the viruses, you end up with more likely causing an eye infection. And so that's probably what they do. In your body, what do they do? Again, lots of things, and we're not sure. We just know that they're there, they're doing something, and it's an area that virologists now want to begin to explore because viruses have a bad rep, and virologists need to say, look, I'm doing a good thing. We're learning about things that are important. And so they want to spread into areas to try to understand what viruses are doing that don't seem to be causing a problem. And therefore, it's fascinating. From my background in critical research, in fact, there's no way I could have done any of the work I did at the medical school without viruses. Viruses were central to doing research. And so, take away viruses, I don't know what I would have done there. I guess swept the floor, clean out the cupboards, I don't know what, but they're critical, viruses are. And now we are more and more learning the functions of viruses, and we realize that the vast, vast majority are harmful. Why are some cause disease? Again, they're mutated. Something goes wrong, and that's why they cause disease. And that's what we are now looking into. And so, let's see. Oh yeah, genetically modified foods. Many foods that we buy are genetically modified, and we don't know it. In fact, they often don't say they're genetically modified, even though they are. Seedless grapes, seedless watermelon, the big apples. So many of those foods, that's how they got the big apples, or these foods that are special in some way. They did it by using viruses for genetic modification. But a major function, again, is they keep the bacteria numbers in check. The bacteria just divide. If they have water and some nutrients, they just divide endlessly, unless they run out of space, which is often what happens. And so therefore, viruses keep bacteria in check. And the gastrointestinal tract is plush with viruses. And again, we don't know for sure what they do. Probably they reduce the problems that bacteria could cause. So that's probably what they do. But again, we know that if they're not there, you got a problem. If they're there, you don't have a problem. So it's hard to do research. So they're the first line of defense against invading bacteria on viruses. Plants, the same thing. They allow many varieties of plants to be produced. Move on there. The basis of the marine food chain. If you take a glass of water, seawater, just an ordinary glass, there's about 150 million viruses. And so now we know that viruses are at the bottom of the food chain. So all these, the amoeba and other small creatures, they subsist by living off viruses. And so virus is the first level of the food chain. And therefore, for all these other animals to live, they have to be there. If you didn't have viruses, the whole food chain would be demolished. So that's another reason why viruses are important. Okay. Health. The problem is, again, mutated viruses. Coronavirus is a good example. Thank God for viruses. So when you pray, make sure that you thank God for creating viruses, because they are really the unseen workers that work 24/7 and don't ever complain. They don't demand higher wages. They don't go on strike. They just do their jobs and continue on forever. Coronavirus, I could mention briefly about that, was probably the best response was protect vulnerable and let it spread among young people, healthy people, and you'd have herd immunity. It would no longer be able to infect the population. Generally, when you have 30, 40, 50% of the population has a resistance to the virus, generally it stops it. Herd immunity is something, when you study microbiology, it's a major topic. And so at least 40, 50%, you have herd immunity. And so what we did, I think, for the coronavirus is we prevented herd immunity, and we caused a lot more problems. If we would have facilitated herd immunity among the non-vulnerable, we would not have had the problems that we had. So we basically delayed it. We delayed the inevitable, and now most people have herd immunity. When they check randomly people, they find a large number are resistant because they have had the virus and thus have antibiotic resistance against the virus. And there's the lambda-virus. You can see we know every single gene that's on there. We know what they all do. So they are so important that we've tried to find all the genes. There is a movement now on the internet where people deny the existence of viruses. And we had a friend who was a nurse at our church who argued and argued and argued there's no such thing as viruses. And I said, come on in the medical college, look at the electron microscope, and you can see viruses. Well, that's something else. It's not a virus. But we know a lot about viruses because we've been doing research for many, many years. So with a small group, I can take questions, which is often the best part of a presentation. So any questions? Go ahead.
We've been in the habit of getting a flu shot every year, and the same thing is being said that we should get coronavirus shots every year. Is that a valid?
We have a lot of experience with shots for flu. So we have 40 years experience. So we know they generally work, but not always. The reason they don't always work is because what they do is they go to China, where these originate, and they should take 5 most likely strains that are going to spread throughout the world. So they develop the shot to deal with those 5 or sometimes less, 3. Sometimes they don't get the right strains. So you get a flu shot and you still get the flu. It doesn't have any effect. So they have to get the right strains, at least 3 to 5. Then they Basically, they give you a shot, and your body thinks it's the live flu and responds according to the virus, and therefore develops an immune response. Because it thinks, it's like a fake soldier. Can't do any harm, it's fake, but it responds, the body responds by developing protection against it. And as a result, you end up with protection. So that's basically what they do. That's pretty simple. And was discovered back in smallpox when they started doing research on how to deal with smallpox. That's what they discovered. So that's been a couple hundred years. The flu virus, the coronavirus they're using now, which by the way is the modified flu virus, the problem with that is that they're using innovative techniques like mRNA. And there's a lot of concern that that's not responding properly, that's causing a problem. And partially because we don't really know the effect of that response. And another issue is that I have several friends who were 25, athletic young men, had heart disease and disease of the heart atrophy, and they were dead within a week or two. A friend of mine was a medical doctor, heart doctor, and his son, who was 24, I think, died 8, 10, 12 days after he got the flu shot. So my friend was convinced that the flu shot did it. It's uncommon, but still, in my opinion, at 25, you're healthy, you don't need a shot. You don't need an HIV shot, or a coronavirus shot. So there's no function, there's no purpose. And
So on the subject of corona, but I don't want to throw it off, but ivermectin is an antiparasitic, and it treats it really well. If you look at science research from Australia and different places.
that's what I've read over and over, but some people, when they put that on Facebook, end up
You get thrown
with
out.
some problems.
Yeah, I was just wondering, that sounds like a good strategy, an antiparasitic, right?
Yeah, and from what I have learned, it does work. It's very effective, by the data says. The problem we've gotten is that science has been seen as a god, and we really believed it was godlike, and we followed it, and now more and more studies are saying we made a big mistake. And so what has happened is science is seen as less a god and more effort to deal with some problem. And so I think the data is coming out more and more now that indeed they made horrific mistakes in responding to in this case, the coronavirus. And that seems to be true. As I mentioned, I have a master's in public health, and right away it seemed to me that they were making a big mistake. But then again, I'm not active in the field. I'm retired, and I spend my time writing books and so on. And so I can't start saying that, yes, you should do this or that. So I thought the experts should give the advice, and now maybe I should have given some free advice. Some people would have been better off if they'd taken advice from me, because it's all basic stuff that I learned in my courses for my Master of Public Health. And as a result, I think we made a huge mistake, and a lot of people died. It's said that more people have died from the flu shot, the jab, than they have from the coronavirus.
Do you have any experience with colloidal silver water? Nanoparticles of silver and.
Yeah, silver is a mild antiseptic. You grow bacteria on a petri dish, and then you put different antiseptics on there, different compounds which kill bacteria. And you put silver on there, and it's a mild antibacterial. It's not strong. A lot of metals are mild antibacterial. Silver is one. I don't think it's a panacea, as some people claim, but it's one way of dealing with the problem. So we come up with these fads. I know people who trust silver in everything. Anything's wrong, take silver. That'll fix it. And I don't think so. Other questions? Comment?
Talk about all these mutations. Are there any that improve the bacteria or virus like evolution does?
Well, that's been the hope, of course, of evolutionists, is that we get some that improve. But the best example is that many bacteria have kind of a receptor, and the antibiotic fits in that receptor. And a mutation which changes that receptor so the antibiotic cannot fit in there can cause the bacteria to be resistant to that antibiotic. The problem is that receptor is damaged, and that bacteria is not as healthy, I guess, as bacteria that has that working receptor. It's been damaged, and therefore in the general environment it doesn't do as well. So that's a problem with a lot of research on bacteria and resistance, and so far there's no case that I'm aware of that indeed the bacteria are more evolved or more improved. Generally they're damaged, and therefore bacteria are damaged, and therefore they're resistant to the antibiotic. Which makes sense. You put antibiotic in someone, you've got lots of bacteria, you're going to find some bacteria developing mutation which cause them to be resistant to that antibiotic, like penicillin, and then they're going to grow like crazy. And that's what happens. Just use bacteriophage and you don't have to worry about that.
What
if you
about-
can use it. Go to Russia or Poland.
So how do you ask for bacteriophage as a treatment?
You can, in certain pharmacies, you can buy it.
Compounding?
Compounding, or some health food pharmacies, you can buy bacteriophage. And I'm not sure how they keep it alive until they inject it in your body, but I've never used it. But
Why do you think treatment of cancer is so elusive?
that's a whole other PowerPoint. That's a workout
Okay.
I work in. A lot of misunderstanding about cancer. Huge misunderstanding. And actually what the therapy does. It damages cancer cells and normal cells. But normal cells can fix the damage, hopefully. Cancer cells can't because they're damaged. And they, therefore, are more apt to die. So it's kind of a game where you radiate all the cells. If you're healthy, the healthy cells will repair the damage. The cancer cells will become more damaged, and eventually they'll die. And that's what you're trying to do.
Sort of like chemo, right?
And that's what chemotherapy does, yeah. That's a very simple version of what they do. Cancer is basically where the breaks of a cell are lost. And so the cell divides when it does not need to, because the breaks, like P53 is the main one. P53 is the guardian of the genome, and if P53 is working, what happens then is it checks the cell division. If it's all okay, cell division proceeds. If there's damage, it fixes the damage. If it can't fix the damage, it causes apoptosis. The cell is destroyed. So if P53 is working, the likelihood of getting cancer is far less. But then there are other ones of these as well, guardians of the genome, and so it gets more complex. P53 is only one. It's the most important and one of the most interesting, but it's not the only one. BRCA for breast cancer for women, BRCA1 and BRCA2 are other examples of the same thing. So cells are really, well, Jeffrey Tompkins knows all about that. Cells are really well designed.
So when can you come back and talk to us about that?
They invite me once a year. So next year I hope,
Okay.
unless I get another. I'm limited in my travel because I have a wife at home that feels her husband should be at home with the wife. And so she's nice enough to let me travel as much, because last week I was in Pennsylvania for 4 days, here for 3 days, 4 days total. And then next week I'll be in Chicago for several days. So I promised her nothing until March. So you got me full slave till March.
Oh, tell her thanks.
And then March I'll be in Vancouver area and Seattle, and they usually keep me busy.
I'm not really sure how to ask this question because I'm not real familiar, but with the genetically modified foods that we're eating, I've been under the impression they're making it to where they might respond to the insecticide or something, and it messes up your gut.
Yeah, sometimes when they modify it, they cause a problem. lot of times modification is simply turning a gene off so it doesn't produce seeds. And so often it's very simple. But on the other hand, it's not always very simple. We're playing around with something really complex, and sometimes it doesn't work, like RNA viruses, mRNA viruses.
So is there like you said earlier, it's hard to know what is genetically modified, and I guess what body controls the labeling? Is that the FDA?
Yeah, that's tricky because they will say non-GMO, and that's not true.
Really?
So there's a certain definition, and I don't recall what it is, but there's a certain definition when you can say it's non-GMO if you have a certain level of modification. Other levels of modification, they can't say it's non-GMO. And there's a difference there, and I don't know what.
That's so tricky.
Look it up, I'm sure you'll find lots of them.
I got some Campbell's soup the other day, and on the
Oh,
bottom
it's-
label it said genetically modified vegetables included, or
So they're telling him, yeah. So they're more modification, or of a different type. Other questions or comments?
Okay, this is a dumb question for all these smart people, but when you had 150 million bacteria in a glass of seawater, did all those, I mean not bacteria, viruses. Are those viruses cannot replicate on their own, which I think is probably a good mechanism for not, doubling all the time like the bacteria do. But do they go inside the bacteria?
They go inside other organisms and replicate.
Okay.
And then they're released.
And then
So they
they-
replicate and they're released then in the seawater.
But do they go inside the bacteria and replicate and kill the bacteria?
Could be
Is that
bacteria
how-
or other sources, just any other creature they can
Okay.
get in and
And that is a
replicate.
pretty good mechanism, I think, for a creator to say, well, this can only replicate it when it's hijacked something else.
And you got to start somewhere. I mean,
Yeah. Right, that's the lowest.
so that's at the bottom. So all life depends upon that bottom level. You take that bottom level away, and guess what?
Yeah,
Wow.
We don't have the progression from
Yeah.
viruses to whales.
yeah.
So that's true, and that means that the animal can just open its mouth and just swim through the water and just takes in all these nutrients,
Yeah.
breaks down the virus,
Well,
and then
what's
uses
in
parts.
the virus then? I mean, and what kind of, carbon atoms, what is in there?
Well, the capsule might have some carbohydrates on that. It depends on the virus. Some have carbohydrate
Yeah.
as a covering, and it depends. There's many different types of viruses, hundreds of types of viruses, and they all look like they were designed. They're really incredibly beautiful creatures when you look at them through a powerful microscope. Robots. Some look like Christmas tree ornaments. Some look like tubes.
Yeah.
So they're
Well, would evolutionists ever think that a virus mutated or, because you can't get life from non-life. Would they ever think, oh, well, a virus became a bacteria?
what the common theory is that higher forms of life de-evolved and became viruses.
Oh, come on.
So how else do you explain it? And in trying to understand where viruses came from, that's the best explanation they come up with. These higher forms of life de-evolved into viruses. Could you think of anything else? He couldn't. He was an evolutionist.
Yeah.
And he couldn't think of any way that bacteriophage could evolve. No way, because he knew all those parts had to be there, and not just, one leg, it had to have all legs to do what it did, and without all those parts assembled as a unit, it had inability to do its thing.
And it can't replicate itself.
No,
Without it other-
it's got to get inside your cells.
Right. Wow.
And so that's true.
Do you speculate on what you think might have happened at the fall with all the good viruses, and now we got some bad ones? Is that results of the fall? Do you?
Well, we know a lot of problematic bacteria would not be here if it was not for mutations. Going way, way back, from what we know, there would have been no mutated forms of E. coli. So it's pretty obvious that was a form, in fact, fairly recently, evidently, because you look in history and try to find evidence for these diseases, and you don't find, now it may be for a lot of reasons, but you don't seem to find any evidence. And they're concerned about that because they want to know when this virus mutated to cause a problem. And that's one thing that they do to try to understand what happened when, in order for this virus to change so it caused a problem, or this bacteria change to cause a problem. And so before that, it's logical going way, way back, you wouldn't have any bacteria that were mutated. They all would be functional and doing their job like they're supposed to. It's hard, though, for us to accept the idea that these living creatures are important living on us. We just don't think that bacteria could actually be helpful and made by God for a good purpose.
So was corona actually a virus then? Or is it
It's a coronavirus is a virus, right?
Okay.
And it's a class of virus. It has a corona shape, so it's a class of virus. So it's a one of many. And there are thousands of viruses, so in my work we only focus on a few. I did a whole chapter on viruses when I was teaching, and we just covered 12 or 15, and there are thousands of other viruses. Again, that's a fertile field if you want to go into some research field, learn about viruses.
But you really can't do a vaccine against a virus in a normal way.
Well, yeah, you're priming the immune system to respond
Except
to
for flu, I guess you do, but,
killing the virus, yeah.
yeah, but you never, you don't take a cold vaccine. I mean, that's what I've heard anyway, is it changes so much that it would be different all the time.
Right. The problem with many viruses is they have a system, kind of like a deck of cards. You can shuffle it around and get different combinations. And so they can shuffle genes around to get different combinations, so you have hundreds of cold viruses, hundreds of different antigens on the cold virus. And so therefore, you can't come up with a vaccination, get them all, because they're just too many. Hundreds or thousands of different varieties. So what they do is when the virus develops, then they try to catch the latest ones.
Yeah.
And hopefully they get it. And maybe they don't. Good group, so glad that people from ICR came by.
Yep.
So it's good to...
I don't know if you all know, but Eric Hoven's here, and he's in Croatia today, and he's on Twitter. I follow him. I know, and other places, I think.
You're not one of those atheist guys that I have to deal with, are you?
I get your emails, I think, too.
Good.